In this video, we will discuss about impaired T-beta receptor glycosylation impedes TGF-β1 effect. In the previous video, we have seen that there are two N-glycosylation sites in the T-beta receptor extracellular region, and when researchers mutated those two glycosylation sites, they are able to see that the T-beta receptors that are formed has a size similar to the case where T-beta receptor 2 is treated with PNGaseF, indicating that T-beta receptors 2 is non-glycosylated when the two extracellular region sites of the T-beta receptor 2s are mutated. So indicating those two sites plays an important role in the glycosylation of this T-beta receptor 2. Now they wanted to see if these two sites are mutated, will it affect the TGF-β or not? Will it affect the TGF-β related functions or not? So first they check where this T-beta receptor 2 are localized in the perinuclear region. Once these two N-glycosylation sites in the cellular region of the T-beta receptor 2 are mutated. So here the results are shown that for the wild type and for the single mutants, they are most of them are found to be on the cell surface levels. That is, single mutant are still able to give enough glycosylations to the other site that they are able to translocate themselves into the cell surface. That can be seen from this panel 3A and also from the panel 3A dash. So from the 3A dash it is very clear because the 3A dash is for the effectin and also for this flag tag. However, when they check it for this double mutant with this ER marker, that is PDI is one of the ER marker. So when they check it for double mutant, they are able to see that PDI signals is overlaps with this T-beta receptor 2 signals, and this indicates that the T-beta 2 receptors, once they are non-glycosylated or two sites are mutated, they are usually confine themselves into the endoplasmic reticulum, and one of the reasons could be because of the ER stress that is present in the endoplasmic reticulum that does not allow unfolded structures to pass through the further processing or further post-translational modification in the Golgi body. So that could be one of the reason why they are able to see the impaired T-beta receptor 2s are localized in the ER, that is endoplasmic reticulum. So this ER quality control part is not mentioned in the paper. This is based on my own understanding about how the ER works. Thereafter, they checked downstream processing, whether this weakened or impaired T-beta receptors to also affect the downstream signalings or not. Since they are not present the enough amount of the cell surface compared to the single mutant or the wild type, it is expected that their downstream signalings will also be affected. So when they check it without TGF-β, they are not able to see a very high signal for this downstream signaling, that is phosphorylated SMAD 2 signalings. However, when they treated with this TGF-β1 for 30 minutes, they are able to see significant signalings for the wild type and slightly lower signaling for the single mutant, and very low signals compared to the other three cases for the double mutant, indicating that these double mutants also affect the downstream signalings that is reflected in this downstream signaling pathways, that is phosphorylated SMAD 2 pathways. Thereafter, they checked this TGF-β2 relative expressions using CAGA luciferase reporter, and for the wild type mutant, for the wild type and the single mutant, they can see the fold change is statistically significant compared to the case where they are not TGF-β treated. And however, when I check it for the double mutant, although the results are statistically significant with and without TGF-β, their values are significantly lower compared to the other three cases. So this also indicates that the double mutants will cause cause reductions in the T beta receptors 2 formations, because most of them are confined in the ER stress, or most of them are confined in the ER, and later on they could be gone through this ERET, that is endoplasmic reticulum associated degradation, which prevents T beta receptors to expressed.